Later videos will touch on the cutting edge of neuroscience. Before we get there though, we need to go over the basics. The first idea is that function can be located. The frontal lobe contains dopaminergic neurons making up systems of reward, attention and risk. The parietal lobe and occipital lobe going backwards are involved in somatosensory sensation and vision respectively. The temporal lobes at the sides are involved in memory. We know this after years of observation. Like if a patient comes in with the occipital lobe damaged, there's almost always associated vision problems. Now neurons are the wires of the biological circuits previously described. They connect to each other with long processes, and the connection point is called a synapse, which is Greek for conjunction. One neuron will release a chemical called a neurotransmitter. This is shown here as the pink ball. The neurotransmitter binds the receptor located on the other neuron. Conformational change of that receptor leads to the unblocking of an associated ion channel. There's actually a lot of different receptors, but this type is the most common. Positive ions rush in, represented by the golden monkey. This makes the surrounding membrane more positive. If we can get enough monkeys in, the postsynaptic neuron activates. Activation is when that change in membrane potential gets carried through the whole neuron. This travelling change in membrane potential is called an action potential. There are no half-action potentials or especially strong ones. You'll often hear it described as an all or nothing event. The reliability of these action potentials is established by an unusual node you'll find cropping up at regular periods along the neuronal process. These are filled with voltage-gated ion channels. Unlike the previous channel, these don't need a neurotransmitter. At more positive voltages, they let more positive ions in. This feedback gives just the signal, and these nodes are called nodes of Ranvier. As an analogy, think about a long-distance communication wire like the transatlantic cable being fit with repeaters. These amplified an incoming signal and sent it out stronger. That's what the nodes of Ranvier do. This leads me to the next fundamental topic, which is neural pathways, as well as to one of my favourite truisms in neuroscience. Those that fire together wire together. Neurons attach to loads of different neurons, and the ones that fire consistently end up having thicker synapses with more neurotransmitter and more receptors. Those don't fire at all lose their synapses entirely. This is at the heart of brain plasticity, and it's a beautiful thing. Last but not least, imaging. MRIs, like the one on screen, work by exposing us to the super strong magnetic field. And in response, the hydrogen atoms in our body start giving off little radio frequencies. The water in our cells is 2/3 hydrogen, and cells are like 70% water. So these radio frequencies can reveal a fair bit about tissues in hard to reach areas like the brain. Functional MRIs do the same thing, but are calibrated for blood flow rather than just soft tissue structure. The idea is that increased neuron activity requires increased blood flow, because neurons carry very little sugar, and lots of action potentials requires lots of energy. They rely almost entirely on blood for glucose. The functional MRI localizes blood flow, and therefore, according to the theory, regions of intense neuronal activity. Thanks for listening.